Hello dear students, now that we are done with the genetic terminologies, let us begin with the procedure of the experiments. Mendel was very methodical. He first considered inheritance of just one trait at a time. For example, tall and dwarf were studied first. He then further studied two traits together, like for example yellow round and green wrinkled, and then followed by the three traits. He performed monohybrid, dihybrid and trihybrid crosses. He started his experiments with pure line, that is, true breeding plants. He made sure that the tall plant gave rise to a group of homozygous tall and the parental dwarf gave rise to a group of homozygous dwarf. He maintained a complete record of actual number of each type of offsprings. He maintained both quantitative record and qualitative record for about 3 generations. He conducted ample crosses and reciprocal crosses so as to eliminate any chance factor. So let us now see the steps involved in his experiments. The first step is selection of parents and obtaining pure lines. Mendel ensured that the selected male and the female parent were breeding true for the selected trait. He made sure by selfing them for about 3 generations. The second step was artificial cross of the selected parents to raise the F1 generation. For the artificial cross, Mendel had to emasculate the flower of the female parent. So what is emasculation? It is basically removal of the anther from the selected plant to be used as a female. So what is artificial cross? Artificial cross is nothing but removal of anther in the female flower and further dusting of the pollens from the male flower to the stigma of the female flower. Mendel crossed many such flowers by artificial pollination and collected the seeds. He further raised the seeds to form the F1 hybrid, which is also known as the first filial generation. As we have already studied, filial means offsprings. Now, emasculation is something that has to be done well before anthesis. That is, it has to be done before the formation of the pollen grains. Hence, it has to be done in the budding conditions. Let us now study few things about the pea flower. Pea flower are papilionaceous, that is, they are butterfly-like. Corolla having 5 petals. The stamen and the carpel are enclosed in the innermost petals forming carina. Further, the carpel and the stamen mature simultaneously and are self-pollinating. There are about 10 stamen. Filaments of 9 are fused together, whereas the one is free. Hence, emasculation in pea flower is very easy. Just open the carina, remove the 9+1 combination by the forceps. Let us now move on to the third step. Third step involved selfing of the F1 hybrids so as to raise the F2 generation. Mendel allowed natural self-pollination in the F1 hybrid, collected the seeds separately and raised the second filial generation. The second filial generation was then selved so as to obtain the F3 generation. Here we are done with the procedure of the experiments. Let us now see monohybrid cross experiment and monohybrid ratio. Thank you.